Hi. Hi. Can you hear me?
Hi, sorry, just give me a moment. I can't hear you. Ah, okay.
Hello.
Yes.
Hello. Yeah. Today I will have a sliders for others. So I come here earlier to prepare my sliders.
Okay. Sorry, how do I say your name? I don't think I've tried it before. I don't want to say it.
Yeah. My name is Ma Jingyi.
Okay.
Ma Jingyi.
Yeah.
I'm a Chinese.
Yeah. Okay. Thank you. Thank you. Yeah, so just I'm earlier, so because I've got a couple of slides to well, not slide, but like I've got something to show everyone.
Yeah, I found that every time you will come here earlier.
Yeah. Oh, okay. We got someone else joining us.
Okay.
Okay, that's great. So are you also from, are you also working with Wendy, is that correct?
Yeah. I'm a Wendy's PhD candidate.
Okay.
Yeah,
So
I'm a new PhD candidate.
Similar to Wilson, right?
Yeah, I will do some experiments follow Wilson because Wilson will graduate.
Right. So I'm also from I'm from UC. This is my actually my second year out of 3 years. So I've got 1 more year to go before I graduate. But I've been working on this since my honors. So I started this project during honors, and I worked on it from a PhD as well. But right now I am also sort of like working with a different part of my PhD, which is chemistry education. So I'm doing surveys and ethics and all that. But I'm also doing this, so it's like half and half.
I will study for my PhD with 4 years.
Ah, okay.
Yeah. And I just start. So yeah, we will have a long time to work together.
Right. Hi, Mickey. Welcome.
Hi, Mickey.
Good morning, afternoon, evening. I don't know. You're in Australia, right?
Yeah. It's 9 p.m. at the moment.
Yeah, 9 p.m.
Is it when do you mention that it's King's Day today for you guys? Is that correct?
Yeah, exactly. Yeah.
There will some activities outdoor. Right?
Yeah, there probably are. I mean, normally there are always lots of events on King's Day, but due to COVID, I don't know like how many there are. But I know like the king is visiting Eindhoven, and they have some activities planned over there. But
yeah.
Hi, Wendy.
Hi, Wendy. Yes, for Australia, we have Queen's birthday, but it's not until actually in June as well. So we still have quite a while to Queen's birthday. But we also get to have a day off on that day. So it's like the important thing is we have a day off. That's great.
Yeah, we have the day off today as well, but yeah, it's only an hour Skype, so it's not too bad.
Yeah. So yeah,
I hope.
Thanks everyone for coming on a national public holiday. It's very high commitment.
Yeah, I hope that the rest also joins, because there was some confusion about the time, because even now it's still display. I clicked on this link, which was displaying 2 o'clock in my agenda. So
Yeah, I didn't realize that. I forgot that daylight saving is ended, so I think there was some issue, but yeah, that's the thing with the calendar invite. It doesn't really take into account of like everyone's daylight savings.
Yeah.
Because there's I think US, I think in the Netherlands you also have daylight saving as well. Is that correct? That happened last year with our meetings as well.
Yeah, so in Europe, everybody, all the countries do daylight saving, but all on the same time.
Yeah.
But in the United States, not every state has daylight saving. Some states do, other states don't.
Yeah, that's like Australia. So most of the eastern states in Australia have daylight saving, but not all of Australia have daylight saving. So that there is an issue, and then it's when I check the time, it's actually overlapping. So sometimes there are like 3 or 4 countries of daylight saving, and it becomes really complicated trying to organize invite everyone to a time, because it's changing all the time.
Yeah, in Sudan, they also don't do daylight savings. So during our summertime, we're in the same time zone, but during our winter time, not. But here in Europe, they had, well, that was before COVID, of course, they were discussing maybe to abandon summertime. But then the issue became, should we all stick to winter time or should we all stick to summertime?
That's interesting. Or we can do a half. We can do like a 30 minutes. That might work as well.
Yeah, but then the 30 minutes is more difficult with the rest of the world.
I see. Yes. I haven't considered that. I think not having daylight savings is also convenient as well, because like you get tired for the first couple of days of switching back and forth. You get used to it, but I mean I get a bit tired. Yeah.
Yeah, but the way our time zone is actually, that would mean that in the summer the sun sets around 9.
Yeah.
In the evening, but it will already be up around 3 in the morning, I guess.
Oh, okay. I didn't realize that. Oh yes, because you're kind of like closer to the North Pole as well, so it's a bit different.
Yeah, so normally in summertime, when it's really summer, the sun only sets around 10 or 10:30.
Wow.
So
Oh,
we had have long evenings then.
Yeah.
But in winter we have shorter days. So
yeah, I think that's what I learned about when I was trying to research traveling to Iceland as well. They have really long days and like long nights in winter and summer as well. So they have midnight sun as well, which was quite interesting. I wish I can see that in my life when COVID is over.
yeah, I wouldn't mind for the summer to have a long day, but I wouldn't like winter to be constantly in the dark.
Oh yeah, true. Oh, you can't win everything, but
And I think it's not feasible to move every half year to another spot in the world.
So where I grew up, which is in Vietnam, it's near the equator, so you kind of just have the same stuff happening every year. It doesn't really change that much. Like you might get one more hour of sunlight, or one fewer hour of sunlight, but it's pretty much just like equal, which is all right, I guess. But having midnight sun would be quite cool to experience once in a while.
Yeah, I like the thing that we have the different seasons so that you really have difference throughout the year. So
Yeah.
Now it's spring. So the trees are getting their leaves now. And then in summer it gets hot. And in the autumn, you see the colors of the leaves change. And in winter, sometimes we have snow.
Yeah, sometimes. We rarely get snow in Sydney, but we do get some snow in the mountain. I saw snow for the first time, like falling snow for the first time last year. I was very excited. That was 25 years of my life. I've never seen falling snow. I've seen snow on the ground, but not falling snow. But it was very.
Yeah, we had snow this year as well, and also ice, so we could go ice skating on the lakes.
Wow, that's interesting. Oh, I think I see Matt joining us as well. And Anthony, welcome. Hi everyone. Sorry, we're just having a chat, waiting for everyone to join us. Sorry for the change in hour, Matt, as well. I actually didn't realize that was happening. That was causing issues. I hope Wilson and Dimitri can join us as well. Actually, not sure if they've got the memo.
Wilson
Well, I sent him
sent a text that he would be a few minutes late.
Okay.
I've got to run at 25 past. Sorry, the change of time is good for a little bit, but I can't stretch the hour. Sorry.
Okay, no problems. I think since Matt is also late, I'm just gonna have to leave early. I'm just gonna give a quick chemistry related update. So I did a bit of what you were talking last meeting about, putting faces to the numbers. So basically putting the structure next to the different different values, so the log D and also the in vitro and in vivo result. So I actually did that. I will upload those files up for everyone to have a look at. But yeah, I haven't had a lot of time to actually do the analysis stuff. I've done a little bit of it, which I'll show everyone in a moment. But yeah, that's what I was doing. So I've got all of the in vitro compounds and in vivo compound redrawn again using the SMILE streams. And I'm just kind of starting to have a look at it. But as Kimberly was mentioning, there's Caitlin who's wishing to join our project as well. She has data warrior, so that could speed up the SAR quite a bit if she does decide to join us as well. So yeah, that's kind of like the chemistry brief update that I have.
Sorry, it's been quite a while since I've been on a mycetoma meeting. As you know, I've poured most of my effort into S2 antibiotics, but I forgot to mention that the amoeba screen does need to be included in this paper. So I don't know if anyone's done that or how far along we are on the paper, but just wanted to remind you all about that.
Sure. I mean, I think we can mention, yeah, it depends which compounds are going in this paper. I guess that'll become clear, but I mean, either way, it'll be easy to slot that in if it fits.
Yeah. Just say something like they were sent to the lab of Chris Rice, tested, not found to be at potent. And it kind of limits the scope of our compounds more towards mycetoma than just generic mycoses,
Just because, yeah, you were just mentioning the interactions with Caitlin, and I didn't have time to follow up on that post, but I mean, it sounds extremely promising. Am I right in thinking that essentially Kimberly and Caitlin, you were talking about involving students from Auckland Uni in the synthesis in the near future? Was that the outcome?
So we haven't spoke with Caitlin about that. That's the University of Washington. So we're doing some out of the lab stuff with them, and then I think it's their fall or summer. So winter for us, summer for you guys. They're looking at, hopefully they can go back into the lab, and they were looking at doing some stuff, synthesis work then. So
For the immunothiazoles or for the phenaramoles?
So we haven't discussed that yet. We're trialing the 2-aminothiazole synthesis starting this week at Sydney. So if that works well, then we'll share that with them. And if they want to have a go with that, they can. If not, I'm happy to chat with them and further discuss where else they could fit into the synthesis. Here's Caitlin now. Hi,
Hi.
Caitlin. This is everybody. We were just discussing the SAR analysis that you were thinking of doing with your students. So, and then we got a little sidetracked. So we might come back to that since you've actually joined us. Maybe you want to introduce yourself a little bit to everybody as well, so they know who you are.
Sure. Sorry, I have to admit I fell asleep putting my preschooler to bed, so I've just sort of woken up again. I'm a little bit not quite with it. My name's Caitlin Berry. I am ex-Sydney Uni, so I know a few of the people here, but I've moved to take up a teaching position at the University of Auckland, and as a teaching focused academic there. And yeah, I worked. I started a new paper last year for our advanced second year students, looking at modelling in the chemical sciences in a really kind of broad sense, and I was hoping to get them involved with this project, because I think that would be a really yeah, a nice in to them for research that fits in with what we're trying to do in the course. So, yeah.
And I think the idea is analysis of data, right, rather than something synthetic at this stage, right?
Yes, yes. So we don't have a lab component with that course at the moment, but last year with my students, we did do a big data warrior project with a series, an in-house series that one of my colleagues was working on. But that project, for a variety of reasons, has wrapped up, so we're not working on that this year. And so I was looking for other, I guess, other projects that we could use the same sorts of skills on, and then this popped up, and I was just like, oh yeah, that's perfect.
I mean, I guess the one thing I would say about that is the that in open source malaria, we had a competition where we highlighted the fact that we had a big data set of activities and no biological target. And we put that out there and formulated a competition where we invited companies and individuals to suggest molecules to make next that were active. And now there's lots of companies interested in doing this. And lots of companies interested in demonstrating that they can do it, publicly in a way that is not going to impact anyone's bottom line. So it's almost an advantage to be looking at a disease where there isn't much money to be made. One of the things that we could do is make sure through doing this work, make sure that the data set that we have is ready for people like that to come along and take it and then use that as a predictive model of what we can make next. In the malaria project, one of the things that took a lot of time initially was getting the data into the right format so that people would just use it in the same way. And the data set that we thought was good was improved further by one of the entrants. So even if that's the only output here, that we have a data set that anybody can rock up to and say, okay, well, I can train a model to predict something that's going to be active and use it to do a generative thing, to come up with a molecule that hasn't existed before, that would be pretty valuable. And I think there'd be quite a lot of interest in doing that.
All right. Yeah. Thanks, Matt, and thanks, Caitlin, for that. So I saw that Wilson just joined us as well. So I think we can officially start our proceeding or meetings according to the agenda. So there's one thing that Kimberly raised, that we do have an issue with our calendar invite. So that's due to the fact that we just having daylight savings for different countries at different times in the year, so that does actually cause a bit of an issue with that. Kimberly, would you be able to just talk a bit more about this if possible? Thanks.
Sure. So basically daylight savings ended here in Sydney and started in other places. And the calendar invites us because they're made by me, they're set to Sydney time. So it's still set to 11 p.m. Sydney time for us. So I'm hoping that we can set a time that now that daylight savings has ended, that's a bit earlier for us in Sydney and possibly a bit later for the alternative meeting. So maybe 8 a.m. instead of 7. So this time slot, I think, is much nicer for us, the 9 a.m., noon, 1, and then 7 a.m. in the US. So if people are happy with that, we can stick with that. I'm not sure, do you still want to alternate? I guess that's the other question. If this time slot works well for everyone, we could just use this same one every fortnight because it's not too out of the way. Before it was, 11 p.m. and 7 a.m. for us. So 9 is better each month for us. Does anyone have any issues with us doing that? If you don't raise anything, I might just create new calendar invites that are set for that time that again are on the final Tuesday of every month.
For me, it's fine.
But I was so just to add on with that, is that going to cause an issue when other daylight saving starts this year as well?
So I think it's something we're going to have to reassess every 6 months or so, right? So just because we're international and times are different, I think when the daylight savings start and end, it's approximately around the same time when they start and end. I think we'll just have to reassess each time. Bummer, but.
Okay. Okay. Yeah, that's. Okay, so the next 4 so is what Matt is saying. So I think that should be it. So sort of like planning for the immediate future-ish. And the other, sorry, I just accidentally muted myself. The other thing is, since we're having a monthly meeting now, it's a little bit, it's a bit more work. Because what we're having is, we need a minute taker. And usually before, when we're doing like the longer interval of meetings, Kimberly used to do that. But if we are going to do a monthly meeting right now, we'll probably need a kind of like a roster of people just taking minutes because it's a bit too much work for Kimberly. So what I propose is, I think between myself, this is just a suggestion. So please feel free to. We can talk a little bit about this. So I feel like between myself, Wilson, Ma, and also Demetrius, where we can start taking turn. So obviously when I post, I can't take minutes at the same time. It's a little bit too much work. But what I'm thinking is we can take turn hosting this monthly meeting. And then whoever is not hosting can then do the minute taking. So that's what I'm thinking. Is I can put a roster if that's okay with with Wilson, Ma, and also Dimitri as well. So that's my plan.
That's a good plan, Hung. But I think I have something to say. This will probably be my last MycetOS meeting. I'm finishing up here. My contract ends at the end of the month.
Yeah.
No, wait. I still have one in May. I'm sorry. So yes, I can do the May if I can do the May.
I think what you can do with Mara is you two can work together for like this, for hosting or taking minutes for these last few meetings that you're here, and then Mara can take over. I think that would be a is that okay with you two and Mara? So I'm sorry I talked over you, but is that okay?
Yeah. Fine.
Yeah.
Cool.
And for Ma, is that okay as well?
Yeah. Can you see my screen?
Yes.
Yeah. Okay. It's Ma Jingyi.
Yeah, Ma, he asked not about presenting your data yet, but if you could take minutes next time as well.
Next time.
So as in so a minute taker is someone who will just write down different what happens in the meeting. So I guess what I'm asking you is it okay if we can take turns? So we can, right now I'm hosting a meeting, but in the future, if it's possible, we can take turn hosting this meeting. So maybe you can be the host of the meeting, and I can
To
take.
be a host of this meeting.
Yes.
Wendy, my English will.
Somebody from our group will take one turn, and yeah, if it will be Ma or me or Mickey, we will see that later. But we will also be hosting or making minutes.
Okay. So that's great. So it's just that we can share. We can just share the hosting and the minute taking. So not one person has to do it all the time. It's a bit too much work for Kimberly at the moment. So that's what I'm suggesting. Is that also okay for Dimitri as well and Matt in? Like,
yeah, we will swap.
Okay. So I'll draft. Yeah, thanks. So I'll draft a rough roster for that, and we'll see to that. So Ma, would you like, since you already have the slide, would you like to start with what you have an update for us as well?
Okay, thank you.
Okay.
Yeah, I'm sorry for that. My English will be improved in the future, I think. Today I will have a short presentation with you all of my PhD plans, and we will start at this picture. Yeah, in the past, our group will we screened the following MMV boxes, almost including three kinds of books, and with 100 to 1,200 compounds, and this led to the identification of 5 proposed compound series, just like that. Most of works are finished by Wilson, and I will do some experiments follow Wilson. Recently, my main job is about the combined response box. Last year, the MMV launched a new boxes, its current box to our group, and this box is about contains 160 compounds with known or predicted activity against COVID-19. So my job is start with this box. We obtained a copy to screen against the 2 common black-green mycetoma causative agents, Madurella mycetomatis and falciformis pora significances. The result is about that. Firstly, I finished the experiments with low and high concentration of the two strains, and the IC50 is ongoing. Maybe I will finish in the middle of May. Yeah. This is recently results of my experiments. The COVID response box with 160 compounds, and the following numbers of compounds inhibit growth is Madurella mycetomatis automatically, obviously higher than the falciformis pora significances. Yeah, we found the 25 compounds inhibited the growth of Madurella mycetomatis and falciformis pora significances. The lower concentration will find the small numbers of inhibited growth. And we did the IC50 experiments. We found 15 compounds from the compounds which have inhibitors both in Madurella mycetomatis and Formispora significances. So we just did IC50 with these 15 compounds, and I will finish the IC50 with Madurella mycetomatis. IC50s, the range of IC50s is 12 to 16, and the IC90 is from 23 to 24, and the IC50 about Formispora significances is ongoing. And the next steps, I will finish the IC50s with Formispora significances, and I will proceed to MIC50 with the MTI50 with the most promising compounds. Next, I will do some experiments in vivo, just to determine the in vivo efficacy against the most promising compounds in Madurella mycetomatis model and Formispora significances model. Yeah, that's all. Thank you.
Wendy, do we have the data for the compounds that were the most promising looking? Just
Yeah,
so
so
I can
all
see what your structures are.
All the data we had is already uploaded in the master sheet. It is under the tab MMV boxes. The only data not uploaded yet is the IC50 because, well, this was very recent data.
Okay.
Yeah, so the screenings are already there. And for Falciforspora senegalensis, I think we need to make a new master sheet. I didn't load it up into the main master sheet of Madurella mycetomatis because I thought it might become way too confusing.
Okay.
The reason why we added Falciforspora senegalensis in these screenings, we also did that with the pandemic response box, is that it's also a very common causative agent in Senegal. But it seems to have a different susceptibility profile than Madurella mycetomatis. For instance, the Fennery moles were also tested by Ma, but they don't seem to work against Falciforspora senegalensis. And in the end, if we do the effort of designing new molecules to treat mycetoma, and we don't have a proper diagnostic test at the moment to differentiate it to really in the endemic countries, it's more safe to go for compounds inhibiting both species. So this was a way for us to see that it would inhibit at least the most common black grain causative agents.
Do the benzimidazoles look good?
It seems that some of them have some activity, but Ma is also screening against those. So what we did was that we, the series we proposed based on our earlier findings, we will screen them as well against this Falciforspora senegalensis, so that we make sure that we know which compounds do inhibit multiple species and not only Madurella mycetomatis. Of course, when we do have a very good diagnostic test, it doesn't matter anymore because then you could treat the best drug for each species, but at the moment it's not realistic yet. So therefore we thought we could also include those species for which no drug is available as well. Because one of the funny things we found actually was that the azoles also didn't work that well against Falciforspora senegalensis, while terbinafine, it was the opposite. So terbinafine did inhibit Madurella mycetomatis not that well in vitro, but it did inhibit Falciforspora senegalensis very well. So, so in Senegal, they most often use terbinafine. So probably they didn't find that different there. But
I was wondering almost if they figured that, I wonder, I'm not sure, did the physicians on the ground there know that, oh, this infection is that if people who have this and it's not being treated by terbinafine, they know is different, or did they just assume resistance?
Well, I spoke to a few physicians in Senegal. Some think that itraconazole works better, others think that terbinafine works better, but in the end, they just simply give the medication which is there. So in some regions, terbinafine is more often used. In other regions, itraconazole is more often used. In Sudan, they always use itraconazole, but they mostly have Madurella mycetomatis. I know that in Latin America, they also have both drugs. And yeah, those are the two drugs in use.
Latin America also has a couple other mycetoma causative agents, if I'm not mistaken, right?
Sorry?
Latin America also has other species other than Madurella and Falciforma spora.
Yes. So in Latin America, you mainly have nocardia, which is a bacteria. So they use a totally different treatment regimen. And we recently also found out that the ones which were supposed to be Madurella mycetomatis in Latin America usually are Madurella pseudomycetomatis. So that's a sibling species of Madurella mycetomatis, but not the identical one.
Interesting.
Can I just ask a question about the units? So those are IC50s and IC90s in micromolar.
Yes.
And I was suddenly thinking about, well, how does that compare with our most active phenoramols? And in many cases, we've been talking about percentage growth of mycetomatis at a certain micromolar of compound. Are we comparing like with like? How do these figures compare with the best in vitro activity of the phenoramols?
So for both assays, we use percentage growth. The assays are similar, so we make hyphal fragments exposed to the drugs, and also the growth rates are relatively similar, although Falciforma seems to grow a little bit slower than Madurella. So in that way, the assays are the same, but genetically seen, the species are very, very different. So it's like human to fish. So the cell walls is probably a little bit different as well. Unfortunately, not much is known about these kind of things of these organisms, but the grains themselves also, they're both black, but in Madurella you see more cement material than in Falciforma senegalensis. So they are different.
Okay. In the master list, we've got a couple of columns in dark green that are IC50s and IC90s. And so essentially we can compare those directly with the data that you just talked about.
Yes.
Great.
So what we also found in mass compound series, the COVID response bonks, the IC50s are relatively high. We had a much lower cutoff value for screening the other boxes. If I know by head, and I look at Wilson a little bit to confirm, I thought we used 8 micromolar as a cutoff for the last boxes. And for these compounds, none of them had an IC50 which were that low. But on the other hand, based on those low cutoff values, amphotericin B, terbinafine were not selected for Madurella mycetomatis, while they do have in vivo efficacy, because we tested them before.
All right. Yeah. Thanks. And thanks, Wendy, as well. Matt has to go. See you, Matt. We'll send you an update. Is there any more questions relating to the update as for Erasmus University? And anyone else has anything to raise? Sorry, was that a question or something? Okay, that's good. So what I think I'll do next is I'll show everyone what I have done well, since the suggestion from the last time, which was, so when Matt was suggesting to just putting structures together with some of these different values, just to see what kind of structures, what kind of chemical structure we have for our different in vitro and in vivo activities. So I've compiled all of the structure for the phenaromals. I haven't quite got to the azo just yet, so it's gonna be for this one just our best hit in vivo, what the structures are, and then how does that compare with the in vitro result as well. So I'm gonna show that now. Okay, so is that okay? Everyone can see my screen. Should be on by now. Okay.
Could you make it a little bit larger, because we can see your screen, but it's very difficult to see the figure.
Yes. Okay. So this is just the Excel. So what I did is from your presentation from last time, I just recreate that graph where you have the in vitro percentage growth at 100 micromolar. So it's this graph here, and then that's correlates with the log D value. So I'm just recreate that here just to have a look a bit more into like the different regions of where things are. So we have like the below, I just took a value below 20% growth at in vitro at 100 micromolar. So that's kind of like these two regions here. And then you have the above 20% growth, which is up here. And I was thinking of just looking at also splitting it between the low log D value as well and the high log D value. So below 2.5 log D and above 2.5. So that's why we've got this purple one, which is below 2.5, and this is above, and then the same thing for that as well. So what I then did is I found all of the different structures. So I labeled them with a number at the front. So that just means I can go back to say, for example, like this one is, for example, is number 56. So I can then go back to whatever is the number in the master list. I downloaded it, so this is not the master list. I haven't changed the master list. This is just my working, so don't panic. I've not modified anything on GitHub just yet. So then I just go back and I found a structure and I found the smart strings, and then I kind of did that for like 200 of them to give me these different structure here that you can see. So and then that just corresponds to these where they are on here as well. And I did the same thing also for the in vivo result. So this is the in vivo efficacy that Wendy showed last time as well, between log D and also the day 10 survival, or survival at day 10. And then I took Wilson's advice, which is the 5 best target or the 5 best candidate for the phenaromals. I think that these 5, which is 5 compounds, so that's Wilson has sent me the structure for those as well. I'm just gonna just overlay them on top of the in vitro result and see how that works. So, oops, sorry, wrong tab. So what happens is when I put together the one that's for the in vitro result that is below 20% growth at 100 micromolar. So I put both of these results together. This is what you have here. So these structures are all, so on the left, like the half left is what's below log D 2.5, and this is above log D 2.5. And both of, like all of these compounds have a in vitro result of less than 20% growth. And then what I also did is because we bring what's the best candidate of our in vitro result, we'll bring that forward, then we do the in vivo result as well. So I colored what we tested in vivo. So the magenta are the 5 best compounds for the in vivo. And then the red kind of like the one that doesn't quite have a good in vivo result on top of it as well. So the red one should be all of the compounds that is below kind of like 12.5% growth at day 10. sorry, 12, like below 12 survival at day 10. Sorry, my bad. So basically the best one are kind of like the purple one that we did in vivo. And then the red one are kind of like the in vivo that didn't quite enhance survival. And then everything in here have been tested in vitro, just to see what kind of structure we're getting out of this. So most of the structure that we kind of already know most of these are very well known. These are our kind of like our starting point. And then when we make modification, a few of those modification structurally, for example, if I modify this, I modify it so that it goes to this, the efficacy kind of goes down a little bit. So the structure kind of like, when you try to modify it, it does actually reduce the efficacy a little bit. So it does have to be quite strict in terms of like what kind of, like for example, this piperazine or this ring here, so this structure has to be like this. If you then switch that, make it a little bit bigger or change the shape, you can see that it will go down in efficacy as well. Same thing if you go from there and you remove a method group, and it also reduce the efficacy. And then I also saw some of these blue ones. These are the one that didn't quite have a good in vitro result. So you can see that they're kind of like 93.5, 85.4, 100. This is the growth, which is on this side here. So you can see if you change that to here, then you actually lose the in vitro result as well, just by a slight modification on this molecule as well. And that is independent of the log D value. So you can see that even some compounds that has really low log D value, which is what we were kind of looking for, if they don't have the right structure, essentially they will also lose their efficacy as well. So it's kind of like a balance between the structure, the ideal structure, and also the log D value. So similar, for similar structure, the lower the log D value is, then you do see sometimes an increase in the in vitro and in vivo result. But then if you completely just change the structure, then even with a low log D value, it won't work essentially. So that's kind of like what I found. So you can also see a comparison between these different modifications. So that's based on this really clear link that we have here. So you change it to a slightly different structure here, and then also change it as well. There's one new thing is I think Dimitri's compound is I think it's the newest addition that has a really good in vivo result. But I saw that there was already another. I think this one is from the MMV box. I'm not sure. I have to check that, but that was sorry, that was from, I think that was from the Epichem library. I'm not sure. I had to double check that, but there was also one before that didn't have quite good in vitro result. So when Dimitri resynthesized that, it actually have a much better in vitro, and we brought that forward to doing in vivo, and it also showing quite promising result as well. So other things such as this one here, this compound here, which is the triarrow, so the free aromatic ring. So if I change the middle bit, so I change the alcohol to another structures like the methoxy here, or there's another one somewhere here, or this structure, you can see that the efficacy will go down as well. I think the alcohol is in the core for this structure anyway is the best. So these are some of the modifications that I made myself. So I do know, a little bit more about them. But I think it will also be really good if Dimitri can have a look at this as well to see in terms of like his modification to this structure, what kind of result you would get as well. Because I'm not quite as familiar with the different compounds that Dimitri make and the kind of the idea behind, or the reasons behind it as well. But I can tell from my own synthesis that this is what I observed. Then I also put, so these are kind of like the one that has the best in vitro result. I just overlay the in vivo result on top of that. And then there's also like the middling one as well. So these are like the growth between 40 and 80% in vitro at 100 micromolars. And also the structural for this is above 80%. So these are the kind of the worst in vitro results. So just from a top down, just overview. Most of these quite large structures, so they don't have the correct structure of what we would expect from those first one that we saw. So you can see, for example, this ring here, there's an extra ring on top, like extension from what we're expecting as well. So you can see if you go to this first structure here, this is like the best one, and this is the substituent or another bit beside it. If you make it too large, for example, like all of these are quite, it has an extra ring, so it has something that makes it a bit more bulky or larger, then it also actually just completely reduce the in vitro efficacy as well. And that is regardless of whether, you know, the log D value is 1.85 or like really low for this one, or the log D value is a bit higher. So in this case, the structures do play a role if it is not the correct or the ideal structure. And yeah, you know, the log D won't matter that much. So that's what I kind of found.
Can I ask a question?
Yes.
Does anybody know how the Fennery moles bind into the binding pocket of the CYP51 enzyme? Because for the azoles, I know that itraconazole and ketoconazole do work well, at least in vitro. They work very well, and posaconazole also works in vivo, but they are very large structures as well, and they are also supposed to have the same target, CYP51. So do the Fennery moles bind differently in the binding pocket or as the azoles?
We would have to get crystal structures. It's thought that they do chelate the iron in there, but I'm pretty sure they would bind to a different site, but they would chelate the iron still. We would need crystal structures. Does anyone, or computational, I'm not sure. Does anyone know someone who could do really complicated docking maybe?
I know
I can
that
inquire one person from our group.
Yeah, we definitely have people we could collaborate with, but it'd take time to set those up and chat with them about it.
But that would be useful because it kind of elucidates like the mechanism of action. And then we can compare.
We know the enzyme target, but we don't know how it binds.
Yeah,
I know that are fungal CYP51s, that there are structures available, and I know that they have computed the Madurella one on top of this, but I'm not an expert on modeling, so I know that they did that, and I know that one of our own PhD students who's not in this group also checked that with a mutation we saw in the enzyme itself to see if it was in the binding pocket of the azoles. But yeah, that was a very, very simple and rough experiment. Our group has not the ability to do this very structural wise or in depth.
We'd have to get a collaborator.
Yes. So anyways, that's what I kind of like initially found. So first I'll upload all of this to GitHub. So if particularly Dimitri, if you have a look, if you want to have a look at it further, like in terms of like what the SAR for your particular compounds as well, I'll put my result and I'll share it with you as well. And is that okay with you, Dimitri?
Yeah. Actually, I've been doing this like similar work for the report as well, so maybe we can compare like relatively quickly.
Yeah. So I'll put this in a new issue on GitHub. I'll tag.
I have a question. Coming back to the analog that I made and which showed like high potency. Yeah, this one. Yeah. you mentioned that it's in the Epichem library.
I'm not sure if it's the Epichem. I'll have a look. Give me 1 moment, actually.
And I actually don't
So
think that
you're
compound
saying it's
was.
not novel.
It's number 19. 1 moment.
I don't think it was in the Epichem library.
Sorry,
I think it was made
was
by
in
Chris.
one of those other box. 1 moment. This one, this tab.
Because I'm pretty sure like every analog that I did like is a novel compound. So I'm just slightly.
Um,
I'm pretty sure. Yeah, I'm pretty sure. A couple of us proposed that that piperidine over time, so we just made it.
so it has a code of P before the MyOS code. It has a code of P4_C_00.
That was a GitHub suggestion. That's what that meant. That was
Yeah, it's from Ben Perry for the mega thread.
that piperidine compound was proposed by me one time and then Ben one time. So
okay, I see what you mean. And yeah, that's the thing. So I was gonna bring this up a bit later, but for that particular compound, there are two lines with the same MyOS compounds. I was not sure what's going on there. Was that
So it's because it's from the same batch. So I didn't resynthesize it again. I just had like a stock of it. So it's from the same batch, but it's been tested twice.
Oh, okay. I see. Yeah, got it. Oh yeah, and then when I went through these different compounds, I can see that for this compound, for example, this is one of our initial target that we saw. There's a few different entries. I think that's 1, 2, 3, 4 entries of this. But I feel like because this one has a racemate and also an S enantiomer version of it. So I think in the MyOS, is it lost or something? I'm not sure what these 4 are they racemate or are they just S enantiomer, because they're actually 2 forms of this compound that I know for sure.
I think they're racemates mainly.
Yeah, we might need to double check that because we don't want to lose that enantiomeric form of this compound. Because I know for sure, because I just double checked that with my my thesis. So you can see for this compound, there are 2 forms of that as well. So yeah, we'll flag that to double check that is correct. That was just I was gonna bring that up a bit later, and you can see some of these do have a retest as well. So you can see if there's 2 lines that tested the same compound tested twice essentially. So that happens throughout as well. So we do have some data, some extra data for some of the compounds as well.
So if it's the same compound that's been tested twice, my understanding is that it should have exactly the same MyOS number, just that there should be one component of that that's a batch number. So the batch number should change from 1 to 2. So we're not creating 2, because like that one there's 37 and 75, but if it's exactly the same structure, it should be 3702 or something like that.
Yeah, because yeah, that's what I have for example here. I have it here, I have it here as well, and I have it there, which is
okay. So that's an issue that's come up multiple times then.
Yeah, so that's what I was thinking. So like it needs to has a sequential number that goes up instead of like a different MyOS, because the different MyOS, like the 5-digit, that's supposed to register a novel compound or a new compound. I do see that problem propped up a couple of times as well. I might need to.
I thought we introduced a batch number within it. I thought we had that. But that's all right.
We'll work on that. So that's an issue that we'll have to look at. So when I did this, I also noticed on our master sheet online that Wendy was mentioning that MyOS 15 and MyOS 30 was at one time switched over. Is that correct?
Yeah, that's what I found, because I also downloaded a copy when I made the graphs. And because one of the questions was that, of course, we numbered everything, how the compounds were labeled in our lab. So when we received APD 14, we used that number over all the time. But just before I made this, the updated slides for last meeting, I renamed them all to the official MycetOS number. And then I went back after the meeting when you had the question, and I went back to the original slide. 2 compounds had changed because there was an
I
issue.
think we were resolving some sort of like mistake, and then the MycetOS numbers changed. So the scheme that you had, like the MycetOS numbers, might not all correspond to them ones that we have like now.
Yeah. So,
but sure. That's one of the options. What happened?
Yeah. So when somebody changes the MycetOS number to the key to the other compound, please also make a mention in it in GitHub, because I'm not checking always the same sheet over and over again to see if there's something changed. So yeah, that's where that mistake was made. Of course, now I found out, so I can change it in my own figure again. But when we need to redo the numbering to make up for the things highlighted by Hung, when that's done, also notify everybody, because then everybody has to go back to the figures they made to make sure that everything corresponds.
But at the time, like when it was changed, so I did actually message everyone. So I'm not sure. Why?
Okay, I might have.
There was definitely like a message on GitHub, like when we were resolving like the mistakes with Hunk.
That was the thread, so I remember that thread. Yes. Okay. As long as you have a message, that should be okay. I think I have overlooked that as well. But so is this current version the final match between these ones? So MycetOS 15 is this compound, and then MycetOS 21 or 30. It's okay now, is that Dimitri? So there's no more changes after that time.
We'll recheck them again, but they should be correct.
Because I also
sorry, message you again, everyone.
Okay, so
just to be 100% sure.
Okay, great. I also went back to the version history of all of these compounds as well, and it seemed to be consistent since 26th of February. So for this one and for this other line here as well. So yeah, I went back to the version just day by day, and that it seemed to be consistent since that time. Anyway, that's from the 26th of February.
Okay.
And one more thing is there is one line missing for this compound here. So MycetOS 19, a log D value was not put in here, and that was. So I think I also went back to the version history of this line of this slide, and I think Ben might have overlooked this and not put it in because he was fixing it, and I think he forgot to put it in. But we might have to just let Ben know so I can put it in, because this compound has been tested, so you do have the all of the in vitro data, so it's just missing a log D value. Everything else we can probably fill in, but we need Ben for that in vitro, log D value, which was not here since the very first time the log D values were entered for all of these ones, which was on the 24th of February. So might just flag Ben for that. I think that's all from me. Sorry, I talked too much. I will stop sharing my screen now, and I'll let Dimitri have an update as well from UCL, if there is any. Would you like to start, Dimitri?
Yeah, just have a quick update. So I actually don't have any schemes to share today. So all I wanted to say is that like I've been working on the new series on the catalog science, and I already got the first intermediate. So I've been working on the yields as well, but I actually got the intermediate. So next time, like by the next meeting, we will have like the first molecule ready. But probably
what molecule is this? What molecule is this?
That's the Cadoxime, the new series, the Perifenox positive control.
Ah, so you're making Perifenox.
Yeah, the ketone intermediate like is not working very well, like in terms of yields. So that's been an issue as well. But at the moment, I actually got it like already, so I can set up like a reaction anytime.
Remember, speaking of those ketoxine or hydroxylamine type compounds, Kimberly had an alcohol intermediate, or was a chloride intermediate that could have been used for making a oxime as well.
It's the alcohol that I was
The alcohol,
the ketone that we've
Yeah,
put into that.
the ketone, yeah. Do you know if you can
I've
react
been
that?
doing like two versions, the 3-methylpyridine with acid chloride.
Kimberly,
and the other
would you be able to react that with
as well.
O-methyl hydroxylamine?
Yep. So I just need to speak to somebody else because we don't have it in our laboratory, but we have a
Okay.
research group who does have it.
O-methyl hydroxylamine?
I just have to borrow some because there's no
Oh,
point buying
cool.
one reaction. But yes.
Cool.
It's on my to-do list, which is very long right now.
Thank you. That's really cool.
Wait, Kimberly, again, like what are you doing? Like with that substance?
So there's a intermediate that we had that Hung actually was working on, which has the 4-chloro, 2-fluoro, and the 3-pyrazine 2 rings. And then there's an alcohol off the central carbon. And I was oxidizing that to the ketone and then doing a reductive amination. But we can actually we can just have that ketone and do this reaction that Anthony's just talking about there to create this other intermediate that we've been talking about for a while. There's a few suggestions, it's been suggested a couple of times on GitHub and that.
Dimitri, I can send you the structure if you'd like.
Yeah, that would be good. Thanks.
I think that's easier than us trying to explain the structure to you.
So with regards like to that Perifenox, so I got the substrate as well, and I just need to set up the reaction, probably even this week, and scale up a bit like on the yield as well.
Okay.
Also, I've been working on some suggestions like in series 1 phenols, specifically with regards to that active intermediate, like from the latest screen. So I was thinking maybe we should do like some more modifications, but I haven't discussed this with Matt, so probably we will update you on this like next time as well.
Yeah, that sounds great. Okay, so we're just checking for time. We're almost to the hours. I think there is one more or two more items on that. This is the outreach and communication bit as well. Which Kimberly, I think that's from you, is that correct? Can you talk a bit about that as well?
Yeah, so it was actually Matt. So Matt got an invite to, I think it's supposed to be a talk / panel type thing. Wendy's also been in on the emails, and Ben as well. So basically, I think it's like a podcast, and they've just invited us to come along to do a recording for that. I think it's looking like it's going to be the end of May at the moment, but they do them often, so it may be at a different time. But yeah, we've just got an opportunity to share what we're doing as part of open source mycetoma, and maybe get some more people on board who might be willing to help out with some components that we're looking at doing. So maybe we'll find someone that can do this molecular docking stuff, or helping with the crystal structures or something, for example. I really wanted Matt to chat about it more because he's the one who's been contacted about it, but he's had to leave the meeting. So yeah, that's kind of in the go. I think, Wendy, do you have anything else to add to that from what you've been a part of discussions with?
So from the latest discussions, I learned that there were many other webinars on mycetoma. I didn't know that, but so might be that we go for a little bit later time point than the end of May, not to intervene with other topics on mycetoma, but I think we should go ahead with taking this opportunity and focusing on treatment and the drug discovery. I think there was already some sort of layout with what topics would be discussed, starting simply with what are the drugs currently used, what is MycetOS, how can we use open source drug discovery to find new drugs, things like that. I know that Ed Zelstra from DNDi wanted to present some clinical stuff, but maybe it's best to let Professor Verhaal do that, as he sees so many mycetoma patients. I think he's the one who should be able to tell how bad the treatment really is. But he was the one saying, could we postpone it a little bit because there are so many talks at the moment. So that's where I was.
Stories about VFend.
Sorry?
I've heard horror stories about some of the azoles, yeah. How they're pretty rough.
Yeah, so yeah, that's where that part of the discussion ended. That was the last update I had.
Should we just put this as one of the GitHub issues so we can come back to that a bit later? Perhaps Matt elaborate on that as well. Let's put that as a new issue for that. Sorry, I'm just gonna bring back to Caitlin as well. So thank you for joining us today as well. Is this so with the work that you want to do for from your team? Is this something that kind of like suitable from what we've been discussing and you've been observing us today in terms of like what you would like to do? Do you feel like this is a good fit for your project at Auckland?
Yeah, I mean, the first thing, of course, is their second year. So a lot of it for them will be training in methods and ideas and processes and what research is all about. So from their end, it's fine. I think there'll be a little bit of overlap with what you're doing around the SAR stuff, but a really different mode of looking at that. So we are teaching them how to do Sally plots in Data Warrior and how to use that particular mode of structural comparison to sort of aid their thinking process about activity relationships. And actually, I think that could be really quite valuable. I find that shows up quite different relationships than when I do it on paper, actually, for me personally. Anyway. But yeah, that looks good. I think it'll just be for me about the advice that I need from you guys, because they're total novices, ways of paring down the data set a little bit so that they're not getting like totally overwhelmed. I really liked the what you presented, where you kind of grouped things a little bit, and maybe we might just look at one or two of those regions instead of the full set. I think for me, it's just finding ways to simplify the data enough that they don't totally freak out, but still broad enough that they can look for interesting things. Yeah, it looks like it could be a good fit. It's fun.
Yeah, thanks, Caitlin. I think that would be your expertise and what you do would be of great help to us as well, especially since we do have sometimes quite large data set as well. So have something to be able to just automate some of that processes as well. That would be a great help to us. And apart from LogD, we do have if you check our master list, there are other different pharmacokinetics and also these different chemical properties as well, if you want to have a look at that. But the things that we've found to have a significant correlation according to Wendy's findings so far is actually LogD. So perhaps if you want to, you can start to have a look at that first. But then there are also other properties as well if you then want to have a look further.
Just a quick question. Any news about your ring lock from Series 1? How are the experiments going?
The ring lock, so in terms of like in vitro in vitro activities, is that what you're asking?
No, are you doing like any synthesis? Because I noticed like on the mega thread, like you have compounds that you are still like in progress, or like.
Oh, yes. So I will send that to Wendy. I am just very busy doing something else. I have another part of my project that is not chemistry. And I have a deadline I need to fill out. And once I'm done with that, I'll ship that over to Wendy before July this year. So, we can do more on that. But I've done most of it. I just need time to actually send it, which I haven't done yet. Sorry.
Yeah, so July is very, very latest, because last year we had a long summer so that we could do experiments all the way till November, but that was really exceptional. So if the R-Venom moles needed to be tested, rather send it in June than in July.
Yes, I'll send it in May. Okay, I just need some time. I'll do it ASAP. I was just very busy. I have an ethics application that I need to fill in at the same time as doing this as well. So I don't have enough time to do everything. But I will send it to Wendy ASAP or to Erasmus. Yeah.
Actually have quite a few new normals ready, like even for now. So
Yes,
we can
yes.
send like at any time.
I do have some ready to be shipped. Yes.
There are new entries like ready.
So are they already in the main sheet? Maybe it's good to give them a MycetOS number to start with, and then set to be shipped. And when they are shipped, so that everybody can keep track on that, so that we can start with using the MycetOS number immediately, instead of using first other numbers and then get confused what number is what.
Okay, awesome. I'll do that. I'll get to that.
So the one that I was talking about, like they're already in the list. It's just they haven't been tested like in the previous season.
Dimitri, I tagged you on the Katoxium.
Yeah, okay.
You should be able to see it now.
Yeah, I'll check. Thanks a lot.
Okay, so I think that's all of the agenda items for this time. I'm not sure if anyone taking minutes, but I'll get the roster for our posting and our minute taking for up and ready for our next meetings. And yeah, I think that's pretty much it. So is there any other things that we haven't talked about that someone would like to raise? Any other business?
Just one quick question. So the next meeting, I'm not sure if we have fixed the time. Will we go back to time A, time B, or just stick to one?
I think we're just going to stick to this time every month, because this is a nice time. It's not putting anyone out. So we'll just stick to the same time every month. I'll create new ICS files and post them to GitHub for everyone to put into their calendars.
Great, thanks.
Okay, right. Is there anything else? I take silence as confirmation. So
We're all good.
So thanks everyone. I hope everyone has a good rest of the day or night, and I will see you all next time.
Bye, Hank. Good luck with the deadline, by the way.